Hello cell biology fans. Let's talk about the citric acid cycle, also known as the Kreb cycle or the TCA cycle. TCA stands for tricarboxylic acid, and so tricarboxylic acid, tri, right, that's 3, carboxylic. What's a carboxy group? So if you think about the C terminus of proteins, they have a carboxyl group. So C double bond to an O to an O. Tricarboxylic acid. So citric acid has 3 of these groups on it. Hence it's got the name tricarboxylic acid. Makes sense, yes? So TCA. All right, what do you need to know? Well, the first thing you really need to know is that it is a cycle. And when we talk about cycles, right, it goes around. And the beauty of a cycle is that if you stop things anywhere in the cycle, right, you put a roadblock in, the cycle will stop working. So that includes if you don't have the input to the cycle, or you don't have other reactants that are required for the cycle. And what you need to know for my class is what goes into the cycle, what comes out of the cycle. It'd be great if you could draw all of this, but remember, we are going to use the ball and stick, black balls on the sticks model to talk about carbons and follow the carbons. Alright, so if we're going to talk about what comes into the cycle, the citric acid cycle starts with one of the reactants is a 2-carbon molecule. For those of you who remember it's called acetyl-CoA, coenzyme A. So it's a 2-carbon molecule that is the product, so acetyl-CoA is the product of when pyruvate is brought across the outer and inner mitochondrial membranes from the cytosol into the mitochondria. You lose 1 carbon off of pyruvate, and you add the coenzyme A to the 2 remaining carbons, and this is the product of that sort of what I call step 1A. So acetyl-CoA is one of the reactants in the first reaction of the citric acid cycle. Acetyl-CoA is going to combine, well, and I always think this is interesting, with a 4-carbon molecule, and that 4-carbon molecule is called oxaloacetate. And remember, I do need you to know the names of what goes in and what comes out. And so oxaloacetate is really the last molecule in the entire cycle of the citric acid cycle, and it needs to be regenerated every time we do the cycle because it's required for this first reaction. So this first reaction ends up generating a 6-carbon molecule, and I always think this is funny because we started with 6 carbons with a glucose, and now we're back to another 6-carbon molecule even though we went through glycolysis and broke it down, blah, blah, blah. This first product is citrate, or citric acid, or TCA. Okay, and that's where this entire cycle got its name. The first product in the reaction is citric acid. Acetyl-CoA and oxaloacetate are substrates. There's an enzyme required to do this reaction. You don't need to know the name of it, but the first product is citric acid. And if we continue, talking about the cycle, just remember, we're ultimately going to go around this cycle, and we have to regenerate the oxaloacetate. If we don't regenerate the oxaloacetate for any reason, if there's a blockade in the latter part of the reactions or in the early parts of the reaction or somewhere midway, if there's a blockage in the citric acid cycle, the entire cycle stops and everything backs up, just like a car wreck on the 405 like we've talked about in class. Okay? So you're going to, right, if you're going to draw this, you're going to say, okay, here comes my 2-carbon molecule. It's going to interact with my 4-carbon oxaloacetate, and we get that first 6-carbon molecule, citric acid. And the next reactions that occur on this side of the equation, which is not really an equation, is that we go from this 6-carbon molecule to a 5-carbon molecule. And for the purposes of my class, no, I don't need you to know the name. And then ultimately we go back to a 4-carbon molecule. Well, where are the extra carbons going? The extra carbons are coming out as CO2. So you end up losing basically, you can think of it as the 2 carbons that came in acetyl-CoA come out as the 2 carbons in CO2. This 4-carbon molecule that we get at the bottom, all right, is not oxaloacetate. And so there are a bunch of reactions that occur on the other side of this equation where you're moving from one 4-carbon molecule to another. Chemistry is happening. All right, we joke around about chemistry happening. But the purpose of this is honestly to regenerate this oxaloacetate. But really important in this is that we're gaining things that we can use in the final electron transport chain, that we're harvesting the energy. In the first, right, when we're going from 6 carbons to 5 carbons, when you're breaking this bond, you are also getting energy out in the form of NADH. And that happens in both cases. In each loss of a CO2, you're breaking a bond, you're releasing energy, and you're getting some NADH. As this process continues around, right, we know that during the chemistry that's occurring and changing the 4-carbon molecule from one structure to another, we get a little bit of energy out here at the bottom as GTP, and the energy there is stored in its phosphoanhydride bonds. Anhydride bonds. Those are high energy bonds. And another place we get some energy out is as FADH2, and this is an electron carrier just like NADH is an electron carrier. So the things that we get out of the citric acid cycle are forms of energy, and the energy is stored in different molecules. Overall, what came in was acetyl-CoA. Right? Remember, that's our 2-carbon molecule. And it combined with oxaloacetate. And that reaction led to citric acid. And that was, oxaloacetate was 4 carbons. Citric acid was 6 carbons. One cycle of the citric acid cycle, or the TCA cycle, results in the production of NADH, FADH2, GTP. Right? NADH and FADH2 are electron carriers. GTP stores energy in that phosphoanhydride bond. What else came out? CO2s, right? CO2s are released. There's still energy in CO2 in bonds. So technically, right, we're not utilizing all of the energy, but we're utilizing a lot of the energy that was originally in the glucose molecule. If you were able to draw these pictures and understand the input and output and the fact that we are converting energy from the acetyl-CoA really into NADH, FADH2, GTP, and CO2 is a waste product in this case, you will be doing fantastic for an exam. Okay, I think that's about it. Let me know if you have questions.